Welcome to the monthly lecture series of the National Capital Area Skeptics. I'm Scott Snell, the current president, live streaming from the Washington, D.C. area, where it's very windy and gusts of like 40, 45 miles an hour. And the power has stayed on, but if it suddenly doesn't, don't think there's a conspiracy, probably. Power has just gone out, but hopefully it'll hang in there. So anyway, the National Capital Area Skeptics is a nonprofit educational and scientific membership organization that promotes critical thinking and scientific understanding. We've been hosting these monthly events, free and open to the public, since our founding in 1987. Now, as skeptics, we consider evidence-based claims rather than faith-based claims. We want to know if a hypothesis has been properly tested. Our speaker today will be touching on that. Also want to know if the test results and conclusions drawn from analysis have been properly reviewed by other scientists, that is, peer review. We want to know if other scientists have later successfully replicated those tests. We also want to know whether the stated conclusions have advanced our knowledge, specifically in terms of predictive power. Are we able to accurately predict an outcome based on what the tests have shown? Our speaker today is going to discuss a particular set of claims. In particular, ionizing radiation is harmful to human health, regardless of how low the dose rate or cumulative dose may be. His position is that the scientific method I superficially described a minute ago wasn't properly followed. He also states that he has historical evidence in terms of correspondence and other personal papers that explain why the low-level ionizing radiation claim was not only accepted by the scientific community, but celebrated with a Nobel Prize in physiology or medicine, and that this flawed research was unjustifiably embraced by safety regulatory bodies around the world, even to the modern day. He's Edward Calabrese, Professor of Toxicology at the University of Massachusetts School of Public Health and Health Sciences, Amherst. Dr. Calabrese has researched extensively in the area of host factors affecting susceptibility to pollutants and is the author of over 1,000 papers in scholarly journals, as well as more than 10 books. Among many other awards he's received over his career, most recently he was awarded the 2023 Herbert E. Schottinger Award for significant contributions in the broad field of industrial and environmental toxicology from the American Conference of Governmental Industrial Hygienists, 501c3 charitable scientific organization that advances occupational environmental health. Ladies and gentlemen, joining us from Massachusetts, USA, Professor Edward Calabrese.
Well, thank you very much. Appreciate you being here and appreciate the introduction. And the introduction was pretty informative to me. And actually just stimulates me a little bit to share that this is not the first time that I've had to deal with skeptics. And I am one myself. And proof's always in the pudding. And I would tell you that that I have, at least in this area of the historical foundations of LNT, published about 150 papers in peer-reviewed journals, a wide spectrum of journals, so that I would have to be confronted with a wide spectrum of editors and a wide spectrum of editorial boards and reviewers, so that they would be seen by a wide range, because I felt that would enhance the overall credibility of what I'm trying to say. It's not just going into the same journal and the same editor and the same reviewers. In addition to that, I'd have to tell you that when I was interviewed by the Health Physics Society for my opinions on the historical foundations of cancer risk assessment and LNT, I was told to bring my documentation along with me. And I didn't really understand what the nature of the interview would be. I thought it would be approximately 2 hours, maybe 3 hours at the longest, and it lasted 2 days. And I hadn't known the interviewers before, had never met them before, and had not been interviewed within this sort of context before. And so when you're going through it, something like that, even though I brought much documentation with me, you really couldn't stop and show people the documentation, or you wouldn't get the interview completed. However, after it was done, and the editing and so forth of this 2-day process was being undertaken, the president of the Health Physics Society, John Cardarelli, demanded all the time, fully demanded that I provide to him the actual documentation, the letters, the transcripts, whatever I was claiming was the basis of what was said, the physical copy to be provided to him. And so I would have to say that in all but 1 or 2 cases, I had the copies, found them, and provided them to him. In the 1 or 2 cases that I didn't, I knew that I had them, I just couldn't find them. So I had to go back to the original libraries, obtain those copies, and to provide those to Dr. Cardarelli, so that the complete set of demands, requirements, whatever skeptical concerns that they had in their hands, and they could see what I saw, so that they could see if I was misrepresenting, how relevant it was, and then what they did as appropriate for this documentary, inserted them, included them within the actual documentary itself, so that viewers could see what he saw, what I saw, and the people who were interviewing me heard. So I just wanted to share with the viewers right at the start of this, and with the introduction from Dr. Snell, that in fact I've tried to play this game, I think the way you would want the game to be played, and that I hope that this is going to be played out. I'd also have to tell you that during the course of my submitting papers for peer review, that it has happened that reviewers have, I can't say question my integrity, but perhaps their interest, they demanded themselves to see the letters that I was documenting within the paper in the references section, and I provided those letters to the editor, that I presume provided those to the reviewers, that were able to confirm what I claimed were in the papers and what I claimed were in the letters, and that sort of thing. And so there is a process that goes on. I think we're all in search for truth, and it's important that we make things available for others who are in that same search for truth to find, and so that people are fully open. And I'd have to say that when other people see things that you read, well, maybe they find that you made a mistake in what you read. Maybe their interpretation's different. Maybe you change your opinion on it the 4th time you've read it. I mean, yes. things like that happen. But I wanted the audience to understand the process that I go through, and that I do believe it's rigorous, and I do believe it's open, and I do believe in full sharing, and I've done that. And and this is just part of that process as we go forth on it today. Okay. So with that is a little bit of a long-winded introduction, but I felt it was necessary for me to clarify this, given your mantra of or your name of the National Capital Area Skeptics Association. I think that following up on that was probably appropriate. So I'll share a screen right now and let the story begin, if that's all right.
Yes.
I'm trying to share the screen, but it says the host needs to enable me to do that.
Stand by.
Yeah. I think we can do that now. Thank you very much. Mm-hmm. Okay, I think we can start this this story. Okay, the title of my presentation today is The Historical Foundations of the Linear Non-Threshold, or LNT, Dose-Response Model for Cancer Risk Assessment. Now, but it's really a story of how LNT was born and sustained. And to dig down a little bit more deeply, it's really a story of mistakes, deceptions, and failed public policy. and mistakes at the highest level, deceptions at the highest level, and failed public policy by our regulatory bodies, including the US EPA and regulatory bodies across the world that followed the lead of the US EPA on cancer risk assessment. And as it turns out, in terms of cancer risk assessment, it's been around in EPA lingo since the early 1970s. But the concept of LNT really hits the journal literature in 1930. And it's not called LNT, it's called the proportionality rule. And it was created by none other than the Nobel Prize winner, Hermann Muller. And Muller is a big-time player. He has the longest reach in terms of influence when it comes to LNT and its belief. And even though Muller dies in 1967, his reach has not ended. His influence has not ended. This man has had a major impact on the last century and the first quarter of this century. Now, Muller, in my opinion, was a brilliant man, very talented. I've read thousands of bits of correspondence, letters, almost everything that Muller wrote, I have seemingly read. And I could tell when he was a consultant, when he communicated with others, that he was just very detailed oriented. He was a very hard worker. And one reason why he was so accomplished, in addition to, I'm sure, his very great inherent talent, was that he worked extremely hard, an extremely hard worker. And I could sense that he thought more deeply than those he was communicating with. And those people he was communicating with, they were no slouches themselves. They were excellent. He had some excellent peers as well. You know, but sometimes the smartest people, they can make mistakes. And sometimes their motivation isn't necessarily pure. And I think we're going to find, in the case of Muller, that as brilliant as he was, that he made some critical mistakes, and he made many mistakes. And that his motivations were integrated with not only public service, but with what I guess I would call self-serving aspects as well, that can distort one's public service. So in my opinion, the first mistake of what I call current EPA LNT policy really occurred over a century ago, or just about a century ago. And it occurred when Muller was really going through his formative stages when he was a graduate student at Columbia and in the decade that followed. But Muller was a graduate student starting in 1911. And he was at Columbia working under become a famous person by the name of Thomas Hunt Morgan. And they were trying to understand, and you really have to go back to this process. They weren't interested in risk assessment. We're talking about risk assessment today, cancer risk assessment. We're talking LNT. They weren't interested in that. They were interested in evolution. They were interested in how did nature create novelty? How did life arise? How did all these species arrive? What made, humans, things of that nature? But going back to novelty, and where did it come from? And they were certainly aware of the origin of species and Charles Darwin. They were aware of Gregor Mendel's work with genetics. But these two, other than looking at natural selection, there wasn't really an answer for genetic novelty. But they knew of the concept of genes, and they knew that if evolution were to take place, it had to be through some sort of alteration in the gene makeup that would be somehow selected for and passed on. The challenge that they had was, well, how did nature do this? How did nature induce gene mutations such that resulted in all this novelty over all the millennia that we see in front of us today? How did nature induce what they called gene mutations? And so Muller and his band of graduate students working at Columbia were working with flies, fruit flies, in the so-called fly room of Thomas Hunt Morgan, trying to figure out how to induce mutation in these little fruit flies. And so they tried everything. Now, how would you as a graduate student try to be the first to induce gene mutations? Well, this is what they did. They took radon and they tried and they failed. They took x-rays and they tried and they failed. They took every single most toxic chemical that they could possibly use, and they used it in the highest possible doses, short of killing the little creatures, and they still could not induce gene mutation. That's hard to believe in today's language. So many agents cause mutations today. Muller and within this framework with other brainy graduate students working from Morgan was not able to do this in a way that was convincing. But they also saw something else, and this was very crucial in their story and situation, and how Muller makes a big mistake. And that is that over the course of these years that they were looking at things, it became generally accepted amongst the geneticists, the fruit fly geneticists of that time period, that they had looked at about 20 to 25 million fruit flies. It's an awful lot of fruit flies. And they had observed about 400 visual mutations. That's about 1 in 50,000. If you fell asleep, counting your fruit flies, you could have missed one and have to wait another 50,000. It was not very common, and this was a generally accepted conclusion. Now, what this actually meant was that, in fact, Muller came to the conclusion that the genome must be very stable, because nature is not producing many mutations in this at all. And so, but what does that mean for evolution? What does that mean for the process? And so what it actually meant to Muller in this process was that, my goodness, if nature is only producing very few mutations, and most mutations are probably harmful, then the few that are selected for by natural selection, that's going to generate the novelty. But Muller came to the conclusion that if nature had the capacity to repair these gene mutations, then it would further restrict the potential for novelty and for evolution to occur. And so Muller came to the conclusion that for evolution to occur, then there would not be such a thing. Nature would not have built in, within an evolutionary context, the capacity for genetic damage repair. And there lies, in my opinion, the original sin. And we'll talk about that. Why? Because as we know today, Muller was wrong when it came to the fact that the genome is highly stable. The genome is highly unstable. As we're sitting here talking and you're listening to me, many, many thousands and thousands of genetic changes have occurred within your cells every minute within every cell. And yet what happens is that our system is superbly fine-tuned to repair these damages. And actually, if Muller had come up with not just one hypothesis, but two, that the genome, he could have said, how do we come up with this 400 mutations out of 25 million? Well, it could have been one hypothesis. It could have been this stable genome, but it could have been an unstable genome that had great repair. But Muller didn't give himself a chance to do that, to test an alternative hypothesis. So as it turns out, Muller makes the assumption that in fact mutation, which is induced by nature or by man, so to speak, is not going to be repaired. And therein lies the original sin, because it actually 99.99, I don't know how many decimal points over we want to count, these damages get repaired. And he integrates this mistake into risk assessment. And this is, showing, just more or less repeating what I've just said. Very few visual mutations of fruit flies, 400 mutations out of about 20 to 25 million. Seemed impossible to induce gene mutations. The genome is extremely stable. Okay, now Muller, 2 years after his big study that gets published in the journal Science in 1929, he summarizes this work that led up to this perspective and how he writes. You can see Muller's an excellent writer. He's very interesting, man, and he's got a personality. If you're really trying to project into this, and this is what he said in one of his paragraphs. Quote, in the course of this work, animals and plants have been drugged, poisoned, intoxicated, etherized, illuminated, kept in darkness, half smothered, painted inside and out, whirled round and round, shaken violently, vaccinated, mutilated, educated, and treated with everything except affection, from generation to generation. But their genes seemed to remain oblivious, and they could not be distracted into making an obvious mistake, which he obviously means a mutation. Tried like crazy to do this, and it was so difficult to do. And at the same time, it wasn't just Muller's group that was trying to induce gene mutations. They knew that there was a big prize at the end of that for the first group that could, the first person who could be the one who could induce gene mutations. Muller wanted to be first. He wasn't the only one who wanted to be first. There were multiple groups in this country and some outside of this country who were going after the prize, and nobody quite knew how close they were and who was to be first. Now, as it turns out, Muller really wasn't the first, but somebody published a claim 6 months earlier in the Proceedings of the National Academy of Sciences, scientists from New York City. But Muller got the big recognition for it, and we can discuss why that happened. But how all this relates to evolution and risk assessment is our focus right now. So in 1927, Muller eventually does induce what he called gene mutations in an article called The Artificial Transmutation of the Gene. And he was emphasizing gene, artificial transmutation of the gene, published in the journal Science. And he did this with very high dose rates of X-rays with his fruit flies. Now, this is a long time after he was a graduate student. I mentioned to you, he started his graduate work in 1911, and he now publishes the discovery of this in 16 years later, 17 years later. I mean, this shows dogged determination. Focus on the problem. Don't get discouraged. I mean, it's a very, very interesting situation. And there was some, why he became first and how that all worked out are parts of this story. So Muller writes in 1929 that he has, this is interesting and important, found at least one of the natural causes of mutations and hints of evolution. So he thinks that he has found, at least in part, how evolution occurred, and it occurred from radiation, and he's going to ultimately come from background radiation. Living in this world that we do, exposed to cosmic rays, exposed to terrestrial radiation, that Muller thinks that this is one of the underlying causes of why we are here today. The only way, however, that he could make that claim, and he did make the claim, was to assert that linearity at low doses, even he had to assert that linearity occurred at low doses. However, one could believe that, but let's take a look at what Muller did in his actual experiment. Muller, you might say, did he use low doses in his experiment that made him so famous? No, he did not. Muller used very high doses and extraordinarily high dose rates. And in fact, the dose rate that Muller exposed his fruit flies to was 100 million, and I mean 100 million, fold greater than background radiation. So as we're sitting here today, life evolving and so forth, from Muller's, attributing this to background radiation, the dose that he gives is 100 million-fold higher than that. So for him to make that claim, what it means is Muller has to extrapolate over 100 million-fold of dose rate to what the background radiation was. So he goes, it's like asking a weatherman what's going to happen in 50 years from now on some date down in some country. I mean, it's way outside your zone. But he's making that assertion. So for Muller, natural mutations, though scattered and very infrequent, were sufficient for evolution with natural selection taking hold and deciding the fate of organisms. Now, I can tell you that I doubt that anybody in EPA, when they were putting together our cancer risk assessment policy, ever thought back to this original mistake. Okay, ever thought back to this mistake. But that's where it occurs. So it occurs because this mistake one is that the genome is highly unstable. It's not this highly stable genome. It's the opposite. You got it totally wrong. Vast numbers of mutations occur each second of the day, each day, each cell they get repaired. So repair is so rapid that one can't tell there was a mutation. And then Muller, as I said before, offers only one hypothesis. Background radiation induces mutation. There's no repair. Natural selection operates. He needed to propose several competing hypotheses. He fails to do so. Muller's discovery gets so much publicity. He's such a big name so quickly. He's such a aggressive individual. He's smart. He can win debates. And if he can't win the debates, he'll pummel you into submission to ensure that he wins the debate. I mean, whatever it was, the field adopted Muller's view. Now, one other question here is, they adopted also not just his view, but did he induce gene mutation? Because Muller said he induced gene mutation, the artificial transmutation of the gene with X-rays. Well, in my opinion, and it's not my opinion, it happened, discovered after that no, he did not induce gene mutation. He induced mostly massive gene deletions. He was knocking big holes in chromosomes that were then recombining. He was like hitting them with a bazooka. And he thinks he's inducing a tiny change in a gene, and that is what's actually happening. He introduces the term point mutation, which designates a relatively, it's a change taking place within a gene. He didn't know about nucleotides back in those days, how many nucleotides are knocked out, and so forth and so on. But he's inducing mostly massive gene deletions. Many notable scientists, they disputed Muller on the topic, showing significant limitations in his argument. But Muller was, he was so close to winning the prize, he wasn't going to back down. Muller would eventually be disproven on this very point with modern methodologies, once you had nucleotide measurement technologies. In fact, even in 1956, 10 years after he gets the Nobel Prize, Muller admits in writing that he actually did not induce gene mutations, but mostly major chromosomal alterations, major gene deletions, moderate gene deletions. So Muller's great gene mutation discovery really wasn't so great. And yet I would bet today in most undergraduate genetics classes that they still teach that Muller induced gene mutation, probably just continuing the inaccuracy of information. Now, what happens with all this? Actually, several things happen. One is that Muller creates the concept of LNT, but he calls it the proportionality rule. And this was born in the literature starting in 1930 out of a need to explain evolution. Evolution was the cardinal belief. Muller concluded that LNT must be the fundamental dose response for radiation. But the only way that evolution could work was via gene mutation from background radiation, and that required linearity at low dose and no repair, and thus the birth of LNT. Now, and I would want to tell you there were a couple things that were done in Muller's lab to try to convince him one way or the other of LNT. He had actually 4 different activities. There were a PhD student and did a work at very high doses with really trying to replicate Muller's work, but extended to a lower dose range and had 4 or 5 doses in his studies. And we were still exposing the animals to a dose rate that was about 30 million-fold higher than background. And his work appeared to be linear, but he wasn't inducing gene mutation either. He didn't know it at the time. However, but Muller had his, this is interesting. as soon as Muller came back from making his big talk in Berlin, where he showed his data on gene mutation, he was harshly criticized by a number of people, but including his best friend, whose name was Edgar Altenberg, who was a graduate student with him. And Edgar says to his best friend, Herman, how do you know that you didn't induce, how do you know that you induced gene mutation? How do you know you didn't just knock big holes in the chromosomes? And he said, you're going to have to actually show that to a critical outside group of scientists. And so even though Muller claimed to have induced gene mutation, he didn't really have the mechanism. So Muller gets a person from Washington University by the name of Frederick Hansen to come out and do a sabbatical and to try to go at this with some sort of a mechanistic way to infer that there was such thing as gene mutation. And as it turns out, Hansen did work that Muller cited that was supportive of Muller's LNT interpretation. However, years later, now this is like the late 1920s, early 1930s, years later, 1948, I recently get a letter that Muller wrote to University of California at Berkeley scientists all about his great concerns that this gentleman from the University of Washington, Washington University rather, had faked the data, him and his graduate student. And that Muller claimed that in 1933, he confronted them that they had in fact falsified their data. And I have a paper published on this that just recently came out. And as it turns out, Muller's very strong in this opinion, and Muller never comes forward with it publicly. It's hidden. Now, why would Muller hide that? Because he claims he's confronted it. Well, as it turns out, this guy from Washington University left Washington University, and he had become a project officer at the Rockefeller Institute. And he had become, over time, Muller's principal funding person, his grant manager, you might say. In fact, he was. And as it turns out, well, now Muller has evidence that his grant manager, when he was a professor at Washington University, was associated with a process that resulted in you might say, creating falsified data. And Muller strongly believed it. And he never went public with that. But so what was Muller doing during this time period? He kept taking money from the guy, but he would, if he exposed him having been associated with potential scientific fraud, well, that may have affected Muller's relationship with whom? His funding agency. So that was never exposed. I've happened to be lucky enough to find those letters. But there were other things that happened too at this time. And you should know that there was a postdoc student who happens to have the same last name as the person who introduced me, Dr. Snell. This person's name was George Snell. And George Snell was a PhD in mouse genetics from Harvard University. And he, after 2 years at Brown University, contacted Muller, and he wanted to try to replicate and extend Muller's work to a mammalian model, a mouse model that he was familiar with. To his great surprise, Muller said, I anticipated this and I was going to do it myself. I have over 400 cages of mouse cages in my setup over here, and we're going to move on it. Thankfully, you know, it would be great if you came on and do it because I have enough work that I have to do as it is. So this George Snell, he arrives in Austin, Texas in July of 1931, and he attempts to replicate Muller's work. And he does it with study designs and everything approved by him and Muller, does what appears to be an excellent study and cannot replicate Muller's work, cannot replicate it at all with respect to gene mutation. And so what happens is this Snell writes up his paper. He never cites Muller's original work, but the work that inspired him, never cited it, downplayed whatever findings he had, kind of hidden away, squirreled away. Muller himself never, ever cites this guy's work, ever. And this work gets swept under the rug. And that was in Muller's lab. Now, within the last 2 weeks, by some amazing twist of fate, I get a new treasure trove of letters that are written by Herman Muller in and around that time period. And I have a letter written by Muller on on March 21, 1933, okay, March 21, 1933, from Muller to his wife, Jessie, and he shares with her that he has learned that he has been nominated for the Nobel Prize with 6 or 7 votes out of the total of 21, and that he and his old advisor, Thomas St. Morgan, they were nominated together, and he speculated that he, in fact, they both may share a Nobel Prize, and it may happen next year. And he says, don't tell anybody, keep it totally secret. You can tell, Altenberg, but tell him to keep his mouth shut. And so it came to me, wow, why didn't Muller cite Snell's work? Well, it was right at the time that Snell's work is coming out negative. Muller is realizing that, gosh, he's in line for this big prize because of his great discovery on gene mutation. And here's this postdoc now working in his lab with his mice in his study design, and he can't extend the work to mammals. And so Muller never cites it the whole rest of the way of his life, ever, not even once. Now, I have to say also, Muller's work was exciting. I mean, he was the first to do this. So Muller even got his department chair to change his focus and to try to extend Muller's work. The guy's name is John Patterson. So Patterson didn't want to do exactly what Muller did. Muller was dealing with transgenerational reproductive evidences of mutation. So this guy, Patterson, he goes out and does what? He says, I'll take a look at somatic mutations. So he gets fertilized eggs, young larvae, and so forth, and he exposes them to ionizing radiation from X-rays, does a massive amount of work, actually. Publishes initial paper in Science and the journal Genetics and so forth, claims that he induces gene mutation, claims that the dose response is linear, and he's highly supportive of Muller's interpretation. I've gone back recently into this, and I've been looking at this area for a long time, but I somehow had missed Patterson's work. So I went in, got all his stuff, and did detailed analysis. I can tell you on my paper that's now under review, I can tell you that he did not show a linear dose response. He showed a threshold response. I mean, it's gonna come out. It'll be out there in the literature, and people can see what. And I look at this guy Snell. I look at Patterson. I look at this guy Frederick Hansen, and Muller doesn't share these with the audience. Talk about selectively keeping things out to protect your own self-interest, right? And so as it turns out, Muller goes to Europe on a Guggenheim Fellowship in 1932, and he gets to interact with some pretty big physicists. But in this particular case, his colleagues, this guy Timofey Rozovsky, who's a geneticist like him, and then 2 physicists, Zimmer and Max Delbruck, they combined to try to take Muller's thinking and to take his dose response relationships and proposed proportionality rule and to add a mechanism to it. So they took a single hit mechanism based on target theory of physics and used this to mathematically account for a linear dose response on a target gene. And the problem with the model was that it was wrong from the start. And why was it wrong from the start? Really, because it was based upon Muller's incorrect gene mutation conclusion, and where he's knocking large holes in chromosomes. And he also assumes that there's no such thing as DNA repair. So there's no repair component to this model. So when this LNT single hit model was created, I can tell you 40 years later, it was given to EPA, and that's what was the framework for how they started. They had a model without repair. Very interesting. So I can follow this up by saying that, Muller was in the midst of trying to defend himself. And he's got a lot of, a lot of people they're questioning him. And Muller was trying like crazy to prove that, in fact, he hadn't knocked big holes in chromosomes, and he had this concept called the reverse mutation. And the reverse mutation, he was just trying to prove that the gene was still intact, that it was just a small micro change. And if you could change the red eyes to white eyes in the next generation, then maybe in the next generation, if you hit it with radiation, you could change the white ones back to red. And he called that a reverse mutation. And therefore the gene would not have had holes blown in it, but it just micro changes, and that's how evolution would have occurred, so Muller thought. And so Muller had this reverse gene mutation concept, and he was trying to use this so he could show a mechanism, show an understanding of what was going on. And after 9 or 10 years of this, and people coming in from all sides, the vast amount of literature failed to support his position. And Muller was losing ground fast. He had induced transgenerational phenotypic changes, but it wasn't at the level it wasn't gene mutation that they were challenging. And then tied into this whole thing was one of his great rivals, this guy, Louis Statler. He was at, of all places, Washington University, but he wasn't, he knew Frederick Hansen, but it was separate. But as it turns out, Statler was a con geneticist, and Statler was a rival. Statler was only 3 months behind Muller, but Muller beat him to science and getting his publication. So Muller gets the prize and not Statler. Well, as it turns out, Statler meets a PhD person by the name of Barbara McClintock at Cornell University, where she got her degree, and she's a genetics, like a cytogeneticist. She's really trying to understand how you can improve the resolution under the microscope for studying genes and gene changes. She developed different kinds of staining of cellular materials and genetic materials, and she had made some major improvements on resolution by at least an order of magnitude. And so he invited her to take a faculty position at Washington University, which she did. And while there, what he does is he shares with her the results of an experiment with corn, and where he had induced what he called gene mutation. So she applies her new technique to, in fact, the cells of his corn, and she looks at it with her newly highly resolved staining technique, and she goes and says to him, "You think you're inducing gene mutation? All you're doing is knocking large holes in these chromosomes." And he was upset because he thought that he had induced gene mutation. He refused to accept what she said. They argued back and forth. But in about a month's time, she basically convinced Statler that he had not induced gene mutation. So what that did to Statler was, in fact, Statler said, well, if I didn't induce gene mutation, then guess who didn't induce gene mutation? Hermann Muller. And all these things were just stacking up against Muller. So Muller he was losing. I mean, Statler was at least Muller's equal in intelligence and probably better in study design. And he had just Muller boxed into a corner and couldn't get out. So Muller then says, well, what am I going to do? So he comes up with a new concept, and that concept is, oh, maybe I can solve two problems at the same time. I will try to do this in a different way, because I'm not going to win on this reverse mutation angle at all. So he takes an experimentalist view, and he says, well, I believe that I can prove this linearity situation if, in fact, damage is due to a single dose, a total dose. That is, every single dose occurs, none of it's repaired, it's irreversible, the damage is cumulative, and it becomes like a piggy bank theory. And if over time, then, when you look at it dose response wise, it looks linear all the way down to a single change. And he said, as compared to, well, if there's a repair process going on, then you have to exceed the limits of the repair process before you're going to see any change. So he said, I think this total dose thing is right, and we'll test it. So he gets a graduate student in Europe while he's at the University of Edinburgh to test this hypothesis. And it's in the late 1930s. And so, as it turns out, this student does this work. And to my personal great surprise, the student showed support for the total dose rather than the dose rate hypothesis. He had supported what Muller had said. And Muller took this as, whoa, this was God, I got Statler off my back. I have this total dose wins, the piggy bank model wins. It gets me down to almost a single ionization, a linear dose response. And Muller had the student publish it in a conference proceedings, and that revitalized his standing in the field, and I think played a significant role in him ultimately getting the Nobel Prize. It just reestablished him. Now, but as it turns out, I've gone into this dissertation in considerable detail, and also the time period and so forth. The first thing to know is that Muller, when he went over there, the guy was not really trained in Drosophila genetics, and Muller had to teach him how to do strain crosses and many other techniques. And Muller did not stick around long. He was there for a brief period of time, taught the kid what he could teach. Muller comes back to the United States for a long visit and doesn't return until the research is totally completed. So Muller wasn't there when the dissertation was done. Now, I've obtained the dissertation. I have the letters between the student and Muller. I've obtained the documentation for the graduate advisory committee and the criticism of the dissertation. And things that you learn here of what a flawed study it was. The study was piloted with a strain of Drosophila that wasn't tested. And then what happened was that he was terribly inefficient and incompetent, really, for the level of performance he needed to come up with a single strain. And he came up with multiple substrains. And when he tested his animals, and I have the substrains in their relative proportions, he has different substrains of bacteria proportions in the controls versus the other treatment groups, introducing other variables into this. The mix of strains that he used could not differentiate between mutation, could not measure mutation and translocation effects in the chromosome. So they because it couldn't do two things, they dumped what they were working with and came up with another mix of strains all at the same time, and they ultimately combined the data with the first and second group together when in fact the risk of mutation at the control group level between the two groups of fruit flies differed by about 3 or 4 fold. They had so many problems. They had failures with temperature control and system failures, and just from looking at it from my point of view, yeah, actually having gone through, having managed many graduate students and having gone through the process myself, yeah, the first year of PhD life is kind of a screwed up year. You're trying to learn what you're supposed to learn, and things get thrown out, and or you revise and you go forth in your subsequent years. This was a dissertation that was to be done in the first year. It was really almost hypothesis generating, and then to top things off, I didn't pick up on this right away, but one morning I woke up at about 4 o'clock in the morning and I said, where were those incubators? Where were his incubators? And this is in this dissertation. So I run into the school, I pull out the dissertation, pull out his papers. I'm trying to find out where the incubators were, the control group incubators and the treatment group incubators. Were they together? And most likely, I think that they could have been. And if they were together, were they protected with a lead shield? Because the gamma rays could actually go through the walls of the incubators, and the control groups could get hit. And my calculations were, given the doses that they used, that if they were located near each other, and even if they had lead shielding, the control group would have received at least 24 rads, a massive dose of radiation. And this was never actually reported in the dissertation or in the papers. And so Muller comes out and he reads this. I mean, the paper comes out. This is never identified. It's identified 70 years later by me. Nobody ever, and yet this was used and when he talks about this research and his Nobel Prize work, he's basing almost everything on this dissertation. It's got so many flaws in it and so much missing information. And the entire field of radiation genetics doesn't even point this out for 70 years. And it becomes a highly dependent upon study. Well, as it turns out, World War II happens. Muller makes it back to the United States, and tries to get settled. And because he's such a cantankerous gentleman, very difficult to work with at times, he can't find a job anywhere, even though he's incredibly talented, incredibly bright. You would think somebody would have scooped him up. Well, the only place that really hired him, offered him a job, happened to be in the town that I'm speaking from, town that I live in, Amherst, Massachusetts. And he gets a job at Amherst College. I'm at 1 mile away. I'm at the University of Massachusetts, right? Well, how do you get a job at Amherst College? Well, one of the graduate students, when he was there, was a fellow who worked in the same lab, who got then a job at Amherst College. And with World War II coming, they needed a little bit older faculty to replace some faculty who were being drafted and everything else that was going on. So Muller gets this appointment at Amherst College and stays in Amherst from 1940 to 1945. And so as it turns out, I have to, just as a side point, 1945 he leaves, and Amherst College won't give him a renewed appointment. But 1 year later, Muller gets his Nobel Prize when he gets a job at the University of Indiana. So Amherst College, they gave away a future Nobel Prize winner with very great insight, I'm sure, at the president's office level. So as it turns out, however, when they it's a great school, by the way. They really have outstanding students, unbelievably talented people. And as I mentioned at the start of this, very talented people can make very stupid decisions at times. And so this is another example. But when the Manhattan Project, we think about this, the Oppenheimer and all this sorts of thing, creating the bomb. But actually, that's part of the story. The Manhattan Project also had a need to evaluate the mutational effects and other biological effects of ionizing radiation. Gave large contracts and grants to the University of Rochester. And there were some great people at Rochester who were expert in mutational studies. On the mouse side was this guy, Don Charles, and on the fruit fly side was Kurt Stern, and they would be in charge of different teams. Well, and they were going to be looking at dose rate when it comes to, and cumulative dose on the Kurt Stern side, and some other approaches by Don Charles. Well, as it turns out, we've never really heard much of Don Charles, and he's got his own life, but I can tell you, he evaluated nearly 500,000 mice, and they had no meaningful publications. It was a disaster in terms of return on public investment. And there's a story to be told within that, but it's true that there was no meaningful publications that came out of the Don Charles side. So nothing happened. And you would have thought that if you're going to extrapolate to humans, I'd like to extrapolate from a mouse rather than a fruit fly. But that's not what happened. And so, but we came to depend on a fruit fly to extrapolate to humans. Now, this was Stern's research. It was highly significant, and it affected public beliefs, scientific beliefs, and national policy. And it's why we have our policy today, in fact, very largely. Now, as it turns out, it was the same sort of study. It was the piggy bank model study all over again, but this time it wouldn't be done by a single graduate student working by himself in a laboratory out in Edinburgh and inadequately trained. This time it would be done with big time money, big time operations, multiple postdocs, support staff, do it, Manhattan Project style at the University of Rochester. And so, so the first study was exposing them all at one time and coming up with a damage. And then the next study was going to be taking that exposure was given all at one time, but spreading that out, titrating it over the course of the animal's lifespan. So then you could see, well, if you gave it in all little tiny doses, would it add up to the same amount of damage if you gave it all at one time? Muller says, yes, it would. That's his piggy bank theory. For those who are in the dose rate side, they would say, no, it's not going to add up. And you may not even see any damage at all. And so, but you'll see a whole lot less. That's what they were really telling. Now, even now, the acute study was done by this guy, Warren Spencer. The second, more chronic study was done with Ernst Caspary, both working under their boss, who was Kurt Stern, a well-known guy and friend of Muller. Well, as it turns out, just picture this. They all believe, they believe like it's a religious belief. All the way tight They believe in LNT. This is the mantra of the radiation genetics community at that time, led by, you know, their guru, Herman Muller. Okay, I mean, it's kind of group think. They're smart people, but it's group think. All right. So then you have this postdoc who is Ernst Kasperi, who does this work, and he comes in and he says, my study does not support the acute study that found damage at these, you know, when you gave it all at one time. When I gave that same amount of dose and I spread it out over the entire animal's life, I didn't see any damage beyond what was in the control group. It was a flat liner, so to speak. There was no increase. That doesn't support the LNT. It doesn't support the piggy bank model. It supports a threshold. And so this posed this very serious challenge to this LNT concept. And this is where things begin to go off the rails here. And so what happens is that Stern's a believer and he's tied into Muller's camp. So he says, I don't accept your data. And he said, well, why don't you accept my data? Well, because he said, I think your control group was reading aberrantly high. And if it's reading aberrantly high, then it creates a false data and it'll show a threshold rather than an LNT. He says, we wasted a whole year. We wasted several years on this, but that's the way it is. And so what happened is that they still had to write up their results. And so what's going to happen here? So, well, as it turns out, Kasperi, I'm going to give him credit. He digs his heels in. He goes into the literature. He pulls out a number of studies that have control groups just like his. And he goes and shows that his control groups were not acting aberrantly, that they were acting as they should, right in the middle of the pack, so to speak. He goes and shows this to Stern, and guess what? Stern, the good scientist that he is, actually backs down and accepted the fact, based on not so much a fact, but accepted the argument of Kasperi that in fact his control group was not acting aberrantly. He couldn't use that argument against it. And so, so what happens next? This is where it gets kind of weird. I'm sure with pressure that came to bear upon Kasperi, between Kasperi and Stern, they wrote up their paper. And in the discussion of that paper, guess what? Instead of saying, oh, we have a novel discovery, we've discovered support for a threshold. Wow, this is the biggest and best study to date, which it really was. It didn't have any of the methodological problems of that which came before him. They said, oh, we have data that doesn't show any increase that supports a threshold, but we don't want you to accept these data until we can find out why they didn't support a linear dose response. We don't know why this occurs. We're going to have to try to figure out why these studies differed. And I'll tell you, I read that conclusion and I couldn't actually believe it. And they spent 6 or 7 pages saying why not accept, why you shouldn't accept our data. And I said to myself, well, what journal would ever publish a paper where the authors wrote a discussion and a 6-page discussion coming up with all the reasons why their data shouldn't be utilized? I said, well, the only editor who could accept that might be a person who was a co-author of that paper. And in fact, what Stern did was he submitted the journal to Genetics, a top journal, and he was the editor-in-chief of the journal. So he submits his paper that claims that they can't support the LNT and doesn't know why and everything else, and don't accept our data, submits it to his own journal and doesn't really, he submits them on November 25th, 1948, and they publish it in January 49, 70-page paper. I'm sure it was not peer-reviewed, but this starts them down a road. And I'm saying, well, this did not support LNT. This supported a threshold. Yet I know, because of my reading, I know that Muller, only, a couple months later, he's getting his Nobel Prize. He's speaking on December 12th, 1946 in Stockholm. And Muller claims there's no possibility of a threshold, that you have to switch over to an LNT. He cites this Edinburgh study. And I'm reading this and I'm saying, how did Muller ever state that? Muller was a consultant to a paid consultant to the Manhattan Project. He told them what study design to do. He provided them the fruit flies to be tested. He made trips up there. Didn't they share this data with Mueller? Well, maybe they didn't. I've consulted enough to know sometimes the sponsor doesn't show everything to you. Maybe Mueller didn't know this. So I bought all the literature, all the correspondence that I could between Mueller and Stern, and Mueller and Kasperi, Mueller and this one, Mueller and that one, whatever I could get. And as it turns out, I found that, yes, on November 6, 1946, in a letter, Mueller actually received from Stern the data set and the paper. And Mueller wrote back on November 12th to Stern saying he got it. He's glanced over it. He could see, oh my God, there's a real problem here. It's challenging the linear dose response relationship. This guy Kasperi is a good researcher. I'm not criticizing him. You've got to get more money to test this hypothesis. So I knew that 1 month before he gave his Nobel Prize speech, that he had seen the data that supported a threshold. And that wasn't just any data. It was a data that was from the Manhattan Project that he was a consultant on, and that had more staff support than anything possibly. And also I knew the quality of the work of Kasperi, which was pretty high. And so then, but Mueller goes to the Nobel Prize audience, and he basically, in my opinion, what he does is that he misrepresents the literature to the world audience, misrepresents it. Now, let's say I happen to be Mueller, okay? I'm Mueller. I'm going to get the Nobel Prize for whatever I did. And I stand up in front of them. Now, I'm going to talk about dose response. Am I going to say that there's no possibility of a threshold when I've just seen the data that suggests that? And it's the biggest study to date. I think I would have recommended to Mueller that what you do is you say, well, there's data on the dose response. It's really an unresolved scientific question. It needs further study. There's been one major study out that tended to support a threshold, but I think it's a linear dose response, but the data aren't really in to resolve this. And so we can't go forward with a definitive answer on this right now. More research is needed. I could have told them my opinion. I could have said what the uncertainties were. That's not what Mueller did. Mueller goes in and he actually lies to his audience, misleads his audience. And when a Nobel Prize winner does that in their field, everybody believes them. Everybody believes them. And within a couple of weeks after that, he spoke to the New York Academy of Sciences, and he's claiming that there's no such thing as a threshold. No, no, no. And he continues this mantra. And there are many other circumstances around that. So as it turns out, Stern was lucky. He got more money, and he could try to replicate this work. And then the research actually proved to be even more problematic. And that is they had actually 3 follow-up studies, and these replication studies were problematic. World War II had ended. All the very talented people that went back to their jobs. Stern was forced to rely upon a first-year master's student to replicate the work. It's a very detailed project, and his name was Delta Uphoff. And Delta was really put in a tough situation. While her first experiment, it had very low control group value and aberrantly low control group, 40% below what it was actually showing. And as it turns out, Stern and Uphoff, when they wrote to the Atomic Energy Commission that was funding the work, they claimed that their data were uninterpretable. And why were they uninterpretable? Because they went to Mueller, who was doing tremendous, a lot of work on control group variability in his fights with Statler and other people. He writes to Stern, and I have all the letters, cables, and everything else saying that my data, which is made up of hundreds and hundreds and hundreds of thousands of control group, mutational experiences, they match with Kasperi's, not with Uphoff's. And so this led them to why they called their data uninterpretable. And as it turns out, they didn't ever report this, but I have now published in which I found that they have 2 simultaneous variables in this study. And you can't really do a study with 2 simultaneous variables and claim any cause and effect relationship. It's a fundamentally flawed design at the top, led by Mueller and Stern. And it's not picked up until, my evaluations relatively recently. And so, but nonetheless, even though he claims his data is uninterpretable, and he also claims the possibility that it may have been due to investigator bias, which is probably throwing Delta under the bus. What he does is that he writes a paper for Science again, and it's a one-page paper, a little meta-analysis of what went on in his laboratory, and he goes back to re-criticizing the Caspary work for the high control group, which is totally bogus criticism. He links these other ones together. The data that were uninterpretable now become interpretable. And when he links them all together, he projects this out to be a linear dose response relationship, even though the chronic study data have never been published. The data have been missing for 70 years. They had the control group problem. Whatever, if there was bias, there may have been, there may not have been, I don't know. One would have to have really gone into that in some detail beyond, I think, which is possible at this stage of the game. And they also claimed that in their one-page paper that we'll provide you with a detailed paper with all our methods, materials, and original data, everything, so you can look at everything. And guess what? They never published the follow-up paper. So the study was fundamentally flawed, even if the data were found. Yet, guess what? Nobody in the radiation genetics community ever criticizes this work. Yet this paper becomes a highly cited report used by the subsequent committees of the National Academy of Sciences to provide the fundamental support for the LNT model that they're going to recommend ultimately to the federal government and that the EPA will eventually utilize. So Stern publishes this meta-analysis of 5 Manhattan Project experiments in the journal Science. He now used the uninterpretable data of Uphoff, treating it as normal, reviving the unsupported criticism of Caspary's study, leads to the linear interpretation. It's, as I mentioned, it's a meta-analysis, one-page report, promised to provide all the methodological details in a subsequent report, and guess what? Never does. So in the early 1950s, Muller repeatedly, and this is really weird, inexplicably challenged the Caspary study in writing, claiming that its control groups are aberrantly high. This is hard to believe because I have all the data, all the letters from Muller and so forth to Stern, and his own data from his own lab, which contradict him. And he goes in and he actually lies to the audience. Nobody takes him up on this. Why would Muller do this? Well, this is my speculation. But this guy, Robley Evans, who was a major MIT professor in health physics, he picked up on the Caspary study, and he was highly supportive of it. He wrote a threshold supporting paper to be published in the Journal of Science, sent it to 75 leading health physicists and radiation geneticists. See what I'm doing? Do you have any criticism? All this. He gets overwhelming support. I picked up the letters that were sent to him and so forth in my analysis. And he communicated with Muller on this. Muller got so upset that I think that he wanted to, he had to reassert control, and he was afraid of the impact of Evans. And I believe that's why Muller wrote these 3 or 4 papers in the early 1950s, blatantly lying. And nobody had the courage, knowledge. And I know that Stern and that group would have known that he was misrepresenting the research record, but they never said a word. And so now, fast forward a couple years to 1956, and it's now getting to the closer to the present, past the war, but nuclear is becoming big, and it's going to be nuclear energy. There's the concerns with above ground testing. There's the Cold War crisis, and so the Rockefeller Foundation decides that the government's got to somehow put this nuclear area in some broad perspective. So they write to Eisenhower, and they say, we want you to fund, we want the government to authorize the National Academy to take a look at all things nuclear, medical, atmosphere, let's say oceanographic, agricultural, you name it, 6 different areas. And Eisenhower agrees and authorizes the National Academy to take over and do this. And they say that we will fund the National Academy to do it. So they would provide the money. Now, as it turns out, just so that the audience is aware of what's going on here, the Rockefeller Foundation is going to provide the money to the National Academy. That sounds okay. Now, the president of the U.S. National Academy at that time is a person by the name of Detlef Bronk. Now, Detlef Bronk is also the president of the Rockefeller Institute for Medical Research. So he is a Rockefeller person, but he's also got two jobs. He's president of our National Academy of Science, and he's president of the what's going to become Rockefeller University. He's also on the board of directors of the Rockefeller Foundation. So Rockefeller is giving money to their own person. They're controlling everything. Rockefeller is controlling everything. Okay. I mean, it's a very weird situation that happened, right? Very weird. So what they do is they create 5 committees in which they take technical people in charge of these committees. And then they create a new committee. This is a big point in this story here, a big point. Up until this point, there are a lot of medical panels that have been put together, usually 9 or 12, whatever, how many people you have to them. Usually is 1, 2, or 3 geneticists on those panels. And when they come up with radiation-related questions, risk questions, there's always a discussion over the nature of the dose response in the low dose zone. Is it going to follow a linear dose response or a threshold dose response? Well, the medical community up until that time strongly favored, guess what? A threshold response. The radiation geneticists, they were different. They supported a linear dose response. But if there was only going to be a medical panel, you knew the geneticists were going to lose. They were always going to lose, and they had lost. So guess what Detlef Bronk does? He decides to create a 6th panel and to split out a new panel from the medical panel, and he called it a genetics panel. Okay, he called it a genetics panel. This is a big thing in history right here. It shows you how if you change the rules of the game, you can change the outcome of the game. Okay, it's a subtle thing, but it's more important than the final score in many ways, because it determines the final score. So and now they have been funding, that is the Rockefeller Foundation, had been funding, guess who? Academic research in the area of what? Radiation genetics. They knew everyone who was a major player and what their views were. And the person who was managing all this was a guy by the name of Warren Weaver, who was the director of research for the Rockefeller Foundation. So guess what Detlef Bronk does? Detlef Bronk goes and he assigns Warren Weaver, who is not a geneticist, to chair the geneticist panel. It's the only time here where you have a chair that's not a technical person chairing a panel. All the other areas were chaired by technical experts. Weaver was not an expert, was not a geneticist. He's chairing the people that are the radiation geneticists. And what does he do? He selects only those that have a strong record of supporting what? LNT. Okay, so if you poll the jury before a trial and you say, well, is the person guilty or innocent? Well, I think guilty, 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 guilty. Well, okay, if you're the prosecutor, then I'll choose these people to be on my jury. And you know that it's a stacked deck.
Mm-hmm.
Okay, it's a stacked deck. And that's what the National Academy of Sciences did. And then at the beginning of their meeting, because their meetings were transcribed, and I have all the transcriptions, yes. They read into the record, this fellow who was actually Muller's colleague, Tracy Sonneborn at Indiana at the time, he read into the record the Apostles' Creed of the radiation geneticist. It was all doses cause damage. The damage is not repairable, it's irreversible, and the dose response is linear all the way to a single ionization. And that's right in the beginning of their meeting. Was there any debate over, is there a threshold? Could it be possibly a threshold? What's the data supporting it? No, there was no debate. These people, they were like 12 religious leaders who came to be, and they just knew what the truth was. They knew what the truth was. There was just, let's write down the truth. There wasn't any debate, nothing. And that's how this moved ahead. Now,
if I may just summarize, you stated that flawed science has guided our modern radiation safety policy. And you've given a description about how that flawed science has been implemented, through these, I think you've used the words such as lied, and, that there's conflicts of interest where someone is close friends with the editor of a journal and so forth. In the time that we have remaining, I did want to ask you about, well, hopefully there's recent unflawed science rather than this old flawed science, as you described, that states perhaps that low-level ionizing radiation findings contradict Mahler's findings, and that perhaps this low dose is harmless or even beneficial. I didn't want to short circuit your presentation, but due to time and the ability to ask some questions that I've never seen asked of you, if I may, in terms of the health physics, 22 episodes, I would have loved to have another 15 minutes with you to ask some questions that weren't asked.
Well, thank you very much. I'd have to say that, as you can see, that it's if I lasted through 22 episodes, and I could have actually gone through 44 episodes. There's a lot to the story, and there's a lot more. But the issue that's raised here by Dr. Snell is that, in fact, how do we stand today? What's the how does it? Is the agency thrown off this history, and are they doing it correctly these days? And I'd say, well, actually, they're not doing it correctly today. There's a high reliance upon basically epidemiology to epidemiologic studies with a very high dependence upon the atomic bomb study evaluations that come forth from really dropping of the bomb in 1945. They've been studying this for years. Now, the first thing you should know is that, and this is going on to the present time, but these studies were started in terms of the genetic studies. They were started almost immediately after the bomb was dropped, and they continue now for 70 years following offspring. And the epidemiology is continuing for cancer is continuing. I would say that in terms of certainly on the birth defects, mutagenicity data, and so forth, that are continuing to the present time, based upon the atomic bomb study, that and this is really weird, but James Neal, who was a member of that first Bear 1 committee, he was in charge of the study and brought forward the results of the 10-year study to the group and found that there was no significant treatment effect. They could not detect mutations or birth defects in the offspring of people who were exposed to the bomb, regardless of the dose that they received. That work, to me, I thought that was impossible to believe, but that's what they had. And there was large numbers of people working from Japan and this country on that study, very talented people. That study has been followed all the way to the present time with all new techniques added in, and those data are still negative, okay? Now, cancer they have found to occur, but they have found, now this is also interesting. The first cancers that were found were cancers that were due to, radiation-induced leukemias, because they're an early on occurring cancer. And so, as it turns out, and they were just put forward by a number of people, but Ed Lewis did it first in 1957 with in a science paper. I went back and looked at that data recently and looked at all the data from whatever they were following from early 1950s up for 40 years. And if you really plot the data, you'll find that the data did not support a linear dose response, but a J-shaped dose response. Every single one of those supported a J-shaped dose response. It did not support linearity. And I figured out how he made his error, or his deliberate error, or however it was done. And it was really by taking low dose findings and adding them to higher dose findings, so it would smooth out a curve to look linear at low dose. Others have found what I found and pointed out the hypocrisy or error or mass deliberate confusion by Ed Lewis, who himself became a Nobel Prize winner. But the problem today with public policy is that epidemiology is a great science, but it is not a very sensitive science, and it can only see effects down to a certain level. It has problems with detection because we have so much human background variability.
Yes.
We have genetic factors and other factors. We don't quantify exposure very well. there's one, it's not uncommon in the low dose zone to have one study find an adverse effect and then another study not to find an adverse effect. I've gone through, looked at a lot of the statin literature, on a chemical that's been studied. I mean, radiation has been studied an awful lot. I'll tell you, there's no chemical that's been studied more than statins. You can look at these epidemiologic studies, many epidemiologic studies, meta-analyses of these. It is not uncommon when you get into a low dose zone to find that, well, in this case, well, it was protective in this case. And then the study is supposed to replicate it. Well, it causes an adverse effect in this case because they're dealing with problems with human inter-individual variability, genetic differences. You're dealing with dietary differences, exercise differences, just life experiences, stress differences. These can make a difference within a factor of maybe twofold. And what we're really dealing with is low dose risks, and epidemiology itself is people talk about, well, I want to do now a 1,000,000-man study.
Okay.
Well, the 1,000,000-man study, which is ongoing, that's not going to come up with an answer as well. How good is a 1,000,000-man study? What's the exposure data that you're going to have?
Yes.
How much data you have on individuals? And even in today's world, I can tell you, in the world of biology, plants, fruit flies, and other organisms, you now find that there are impacts that are transgenerational impacts on heredity that are passed on, basically epigenetically, and so forth. And any epidemiologic questionnaire is not going to ask your grandmother what kind of diet that she ate because she's been long dead. It introduces another variable into that process. It's very difficult. So the alternative, it's not that epidemiology is bad. It's valuable. It's essential for humans to have it, but its
limitations very difficult. You've said elsewhere that good faith investigators are on different sides of this scientific question that you've discussed today. Are you able to summarize your quote unquote opponents' one or two best criticisms of your perspective?
Well, I think that on the historical side, it's pretty well documented that, because I'm not really telling my story. I'm allowing Muller and others to tell their own story in their own words.
Yes.
And so for the most part, I'm just organizing their language to tell their story. Where I offer opinions, then my opinions could be challenged and would be challenged, but I try to stay away from opinions and mostly to letting the original authors of this tell their own story, and it falls out that way. But I think that the issue is, I'm really trying to get at the question that's a practical question. It's very difficult from a biostatistical perspective to differentiate at low doses to differentiate a threshold model from a linear model. Given experimental data, limited amounts of sample size, difficulty quantifying exposure, it's very difficult at low doses to differentiate these models. And where this brought me, and I knew this back in let's say the late 1980s, early 1990s, there was an unresolvable conflict. And unresolvable conflict, groups like EPA, they're always going to err on the side of safety. And so what I did for myself was I said, well, I need an alternative model and see if that's a correct model. And so I chose to study for the last 40 years something called a biphasic dose response model, which is a hormetic dose response model, under the possibility that if it were real and if it was true and if it was replicable and all that sort of thing, that in fact that you could have a better chance to differentiate that model from the linear model or even the threshold model. And so my research going down the road, and I've been trying to, you think that the LNT is my major story. No, LNT is my hobby story. My real story for me is the work that I've done on the hormesis side to try to see how valid that is, the quality of the database, how generalizable it is, and can it challenge the LNT interpretation, even the threshold interpretation. Yes. And I believe that it can, and I believe that's where the future of the science will go. And on the other hand, I think that the to me it's still a scientific question. It's an open question, and how at the health physics documentary, I was trying to lay out what the future is in terms of how do you compare a threshold versus LNT versus a hormetic model, and let's see which hypothesis wins.
Yes,
And
I posted a link to those videos for interested viewers, and of course we let them know ahead of time about a month ago to watch those as I did. I wonder if the devil is in the details of what your threshold value may be. For example, forgive my layman thinking about this, but there are some people I know that seem youthful even when they get very old. And then there's some young people that look older than their years. And I wonder, that ability to repair damage of, say, low dose ionizing radiation varies from person to person. So you want to set a threshold that protects the weakest, where people that look older than their years, because their repair mechanism, again, forgive my superficiality, is not adequate to deal with ionizing radiation. And what are your thoughts about that?
well, I'd say this, and that is that I fully agree with you that we have profound inter-individual variability with respect to susceptibility to toxic substances and things that aren't toxic, things that can maybe even be beneficial. So we have profound inter-individual variability. What I may be very susceptible to, you may not be. And there's going to be a wide range of these within the population. Nonetheless, and I have to say this, that when I first got into the hormesis game, I published a series of papers, and this was odd. I published a paper in the annual review, one of the annual reviews, and it was you would think that I think it was the annual review of public health, but when I published it, I've never seen this happen before. One of the people on the editorial boards decided to write a separate article criticizing my annual review, right in that same issue. I've never seen that done to anybody ever. When writing an annual review, you're supposed to be an authoritative person, and here's somebody challenging my views. But it was very helpful to me. His criticism was good. He said, you have shown hormesis in robust populations, healthy populations, and those adaptive responses are okay. You proved your case. But what about the challenge that EPA has, which is the challenge that your question is, and that is, what about people who are compromised? What about the high risk segments of the population? You have to worry about them. There are a lot of that's a sizable section of the population. You can't not think about them. Right. They have to be protected by congressional mandates and things like that. So what I ended up doing was, just to answer your question, was I wanted to see, well, what kind of adaptive responses, in fact, might those who are compromised have? And do they show this hormetic dose response? And I went into my very large hormetic database, and I was able to show that the hormetic dose response, actually, you could induce a hormetic dose response in those who are much more compromised. That even though they were compromised, they still had an adaptive capacity. It doesn't mean that they're at less risk. They're not at lower risk than those who are more robust, you might say, with their potential. But in fact, that even if you are on the low end of that distribution, so to speak, that you still have the capacity to be exposed to I'm going to say environmental stressor agents, whether they be chemical or physical, and that you can actually upregulate adaptive mechanisms and protect yourself to some extent. Now, how that all plays out within a regulatory framework, where it finally drops off, if you have multiple risk factors, then maybe your adaptive capacities drop off. And I would have to tell you that when you get to my age, which is 77 and a half years old, my evaluation of adaptive mechanisms, my hormetic potential is far, far less at my age than it was when I was 45.
I see you've anticipated one of my questions and I felt silly about it, but I wonder if you keep a low-level ionizing radiation source around, perhaps near your bed or something, and if people want to laugh, go ahead, but I am curious about that. May I ask?
Well, let's put it this way. I've heard that recommended many years ago because I run a lot of conferences and know people who were really into that. I
Mhm.
would say that there are, and I spend most of my life trying to find safe ways, not controversial ways actually, to try to upregulate adaptive mechanisms. Okay, to protect whatever few neurons I have left that are functioning in my brain, and and
Yes.
to and to protect the other parts of my body, so I can have a reasonably healthy, you might say, lifespan, and maybe I could live X number of years longer. Put it that way, a few, anyways, right? And so I would say that if it's me and I was to advise the American population, I would say that there are other ways that I would recommend to upregulate your adaptive capacities, your hormetic mechanisms, than trying to utilize ionizing radiation. We're getting ionizing radiation from cosmic
Sure.
and from terrestrial radiation. If you read the studies that try to remove cosmic radiation and terrestrial radiation from the lives of paramecia and mice and other organisms, when you remove the background radiation, they tend not to do as well.
Mhm.
They actually need to have some low level of radiation to optimize their protective mechanisms.
I
And
see.
we are getting some of that right now. I think that we could supplement them, and I try to supplement them with my version of, I'll say either my dietary practices or my lifestyle practices that stress me to certain extents, which I think are optimal stresses
Okay.
for maximizing health.
All right.
But I think that those are relevant questions.
Some years ago, my brother gave me a gift from the Trinity nuclear test site in New Mexico. It's, a gift shop. It's somewhat radioactive, and I don't sleep near it, and I'll keep it that way based on your comment there. Wrapping up here, what would you say in a very summarized way is the lessons learned here? I certainly heard you say excellent people, smart people make mistakes. Flawed science, you claim, ended up being accepted, celebrated, implemented in health safety measures that are still being used today. How did peer review fail? Were the peers too chummy? Was this group of investigators too close to each other personally? Was there a lack of a counter Mueller? In other words, Mueller was a very forceful person. There wasn't a rival to him to say, no, Professor Mueller, you are incorrect, and here's the truth. Is that what happened, or am I mistaken?
Well, I think it's complex, but I think there were beneficial motives and negative motives. For example, in general, there's the radiation geneticist community of Mueller and Stern and others during that era. I believe that they truly wanted to protect humans
Mhm.
from the adverse effects of high levels of radiation, especially pregnant women, things of that nature. I
Yes.
think I think I and I think that they were afraid. As I was a child, I was born in 1946, lived through the Cold War. I had to go through these little tests to see hide under the under the desks in school if there was going to be a atomic bomb dropped and things of that nature. There was a concern with trying to protect humanity, and they were part of that. They were part of that issue. And on the other hand, they were also very interested in their careers, and they were interested in making sure, from an academic point of view, they were almost all academics. Actually, I'm an academic. We live and we die off of our grant money. I can
Right.
assure you of that. We do. And grant, and you're competing with other people that live and die off of grant money. You have to make your story more convincing, more scary, more this and more that. and politicians play off of that as well. And so there's a terrible mix between your public service and your self-interest. And I could see this being played out in Muller's situation. Even, for example, when it all started, back when Muller published his paper in Science, he had no data in his science paper, no data on the most important topic possible. And the journal editor of Science published his paper with no data. Now, how did that ever happen? And now I have discovered how it did possibly happen through a quid pro quo arrangement between Muller and this editor. But when the Stern paper with Uphoff was published in Science in 1949, again, it was not submitted to peer review. There was no data in it. 1956, when they published the recommendations of this Bear One committee in the Journal of Science, it was actually contained falsified information because they were trying to manipulate. They actually removed data so that their uncertainties would not be seen by the scientific community and the general public, so their recommendations could be adopted. I've also presented, not today, where William Russell and Arthur Upton. Now, Upton was a former director of the U.S. National Academy of Sciences. They did a gigantic study at Oak Ridge National Laboratories with the radiation and cancer and longevity that showed no adverse effect, and they failed to publish it. They kept it suppressed until Upton, 35 years later, needed that data published so he could win a case for the defense in a UK trial, and they went and they reactivated it. You have manipulations, personal ethical failings, all throughout this story.
Well, that's inevitable, though, for humans. We're all flawed, and the system is supposed to protect us. Double-blind studies, peer review. The system didn't work here, or it has sort of, because it's just taking a long time to try to make replicatable studies for this very difficult problem. You said low dosages are just very difficult to work with. By the way, of course, Chernobyl, Fukushima, I assume people have tried to assess, and it's like, hey, they seem to be fine so far or not. And we can draw some conclusions tentatively, because there should be vulnerable people in there, in addition to the always youthful looking people, and that we might be able to draw on that.
Well, I'll tell you, I find it really troubling. We have a federal agency, the EPA, that's been in business for over 50 years, right? And they should have uncovered what I uncovered. They have grants program. They have thousands of people who work for them. They have multiple generations of scientists, really talented people. And they have a cancer risk assessment process, and they have no understanding of what their history was. They just accepted what was handed to them, and they have absolutely no scientific curiosity.
Yeah, it's a shame that Muller, you said, died in 1967, and the EPA, I believe, is from like 1970, something like that.
Right.
So Muller's papers and his colleagues still living were available to perhaps give background, clarifications, complete context. And of course, your work doesn't have those researchers. They've all died, to possibly say, oh, you're misinterpreting. And that leads to my final question, which is, how did you obtain these letters? Are you certain of their authenticity?
Well, what I have done is I have gone to where these very prestigious people in this country, thank goodness, are recognized as such, and major repository locations exist. If a person's tapped in and identified as major, they try to preserve their papers to some extent. And so the U.S. in Washington, the Library of Congress, has an amazing set of papers of leading individuals in this country and some other countries. There's the American Philosophical Society
Of Philadelphia.
in Philadelphia. And you can see they try to identify who they think are the big people, and they try to how soon do you identify somebody whose papers that you think you're going to want that are really crucial? Not everybody's significant. And we got lucky with Mueller.
A Nobel Prize winner. So in 1967 and perhaps some time later is when his papers were in custody of one of these institutions or more that you've just described. And it's a shame that it's taken so long, but it sounds as though I assume professional historians are also looking at this with you.
Well, but this is what I found. I found the see, I'm trained as a lab-based toxicologist. I'm not trained as a historian of science. I didn't even know how to how they did their work, and I had to in the last 15 years. I had to kind of like put myself through graduate school, so to speak, to learn their methodology and
Yes.
where they got their information, how they did it. And I developed a profound respect for people in the world of the history of science. They're slow and methodical. They're very thorough. They're very objective for the most part. You can learn an awful lot from them. Why my work has been different is these people, as good as they are, it's very difficult for them to challenge a scientist because they're not scientists. And you know, how do you take on Mueller? How do you take on Stern? How do you take on anybody? And so, my training is that you, on the science side, you take on anybody, and because we've been trained to do that as scientists, and so once I kind of meandered into this field of the history of science, I think I saw what these historians of science might have seen, at least to some extent, but I wasn't intimidated to challenge the scientists.
Still, it's good that you're being peer reviewed by professional historians, whatever their shortcomings you perceive, because obviously you also are, benefiting from peer review as Mueller should have.
Right, and I have to tell you, I send my papers to leading historians of scientists. I ask them, I want them to see what I do. I want the scientists to see it, but I want the historians of science to see it, because they have their own ways of ferreting out things and truths and so forth, and two people can read the same letter and come up with differences of significance. I am sure when I read these Robley Evans's letters that I saw things because of my training that almost no one in the world of the history of science would have seen, because we all connect different dots together depending upon what our training is and what we're sensitive to, and so there's probably more scientists should become trained in the history of science, because they could probably see a lot of things that would be seen in a unique way.
The replication crisis, it sounds like you've claimed that this is a notable entry in the so-called replication crisis, if people want to Google that to get an idea about that rather large category. By the way, I wanted to say your friend Phil Wexler says, enjoyed your talk, Ed. Sorry, I have to sign off. Take care. And Curtis Haymore in Virginia remembers your talk to the DC chapter of the Society for Risk Analysis on low-level radon exposure. This was back in the early 1980s. He thanks you also for your presentation today. And I'll wrap up. I hope you described the difficulty technically of trying to get at the excellent data for this problem. And I hope as techniques improve, apparatus improves, and peer review and the scientific method will clear out this problem that you've described. And I don't think anybody wants to embrace falsehood, although perhaps to err on the side of caution, which I think was a significant phrase in your presentation today. I think that may have, plus hubris perhaps on the part of Professor Mueller. All right, well, thank you again. And I want to thank you, Professor Calabrese, and note that today's program was produced by JD Mack. Thanks, JD. On behalf of the National Capital Area Skeptics, I'm Scott Snell saying so long from Greenbelt, Maryland. Please join us again on Saturday, May 4 at 1:30 p.m. Eastern Time. We'll be in Arlington, Virginia in person at the Central Library there. Our speaker will be Zelda Gilbert, Professor Emerita of Psychology at Woodbury University. She'll present The Psychology of Conspiracy Theories. Our event will also be live streamed here on our YouTube site, but we love to see you in person at Arlington Central Library on the 4th of May at 1:30 p.m. Eastern. See you later. Bye-bye.